Pollen-Pistil interaction. It is the interaction between the pollen and the pistil. Pollination. Pollination is the transfer of pollen grains from the anther to the stigma of a pistil, right? So pollination does not guarantee the transfer of the right type of pollen. Often, pollen of the wrong type, either from other species or from the same plant, also land on the stigma. So for this pollen-pistil interaction, we have several processes or events. So the first step is the pistil has the ability to recognize the pollen, whether it is of the right type or of the wrong type. If it is of the right type, the pistil accepts the pollen and promotes post-fertilization events that leads to fertilization. So that is the first step. And the second step is, if the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue between pollen grains and the pistil. This dialogue, that is the dialogue between the pistil and the pollen grain, is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. For example, let's compare this one. You went shopping to buy a shirt, and you are the pistil, and the shirt is the pollen grain. And when you go, you put on the shirt, and your friends will be there, right? They will give comments. So your friends will the chemical components which mediate between you and the shirt, right? In the same way, the chemical components will mediate between the interaction between the pollen grain and the pistil. Okay. So for you to accept that shirt, you need you yourself, the shirt, and your friend comments, right? In the same way, for this pollen-pistil interaction, for the pistil to accept the pollens or to reject the pollen, it needs the pistil itself, the pollen grains, and the chemical components. Okay. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. Do you still remember what is germ pore? It is the pore or the aperture which is present in the egg sign where sporopollenin is absent, right? And the function of germ pore is formation of pollen tube and release of male gamete, that is pollen grains, during fertilization. Okay. The contents of the pollen grains move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary. So, pollen tube grows through the tissues of the stigma and style and move through the ovary. You would recall that this is the fourth step, okay? In some plants, pollen grains are shed at two-celled conditions. That is, two-celled is vegetative cell and a generative cell, right? So this is the fourth step. In some plants, pollen grains are shed at two-celled condition, that is vegetative cell and generative cell. In such plants, the generative cell divides and forms the two male gametes during the growth of pollen tube in the stigma. In plants which shed pollen in the three-celled conditions, pollen tubes carry the two male gametes from the beginning. Pollen tube, this is the last step. After reaching the ovary, enters the ovule through the micropyle and enters one of the synergids through the filiform apparatus. The pollen tube, after reaching the ovary, will enter through the ovule of the micropyle and will enter one of the synergids. Okay, through the filiform apparatus. What is the function of the filiform apparatus? That is, it will guide the pollen tube into the synergids, right? Many recent studies have shown that filiform apparatus present at the micropylar part of the synergids guides the entry of pollen tube. So all these events from the pollen deposition on the stigma until pollen tube enters the ovule are together referred to as pollen-pistil interaction. As pointed out earlier, pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help in the plant breeder in manipulating pollen-pistil interaction even in incompatible pollinations to get desired hybrids. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, Crotalaria, balsam, and vinca on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grains. So just this much. It's this there. I make it as 5 steps. So just with the 5 events or the 5 process, the 5 steps. So these are all about the pollen pistil interaction.